Welcome to this lecture about DNA replication. So DNA replication is a complex and tightly regulated process, and we will see some of these processes. We will see them relatively in details. However, keep in mind that it is even more complex than what we will cover in this chapter. So let's first make a little overview of the DNA replication process. So first of all, remember that it is performed by a small number of proteins, even if it may look like there are a lot in this chapter, and that they are highly conserved. So it means that the process being the same in every type of cell, the proteins that are involved in it will be quite similar from the bacterium to the human beings and even plants. So even if the proteins are not exactly the same, they have similar functions and similar structures. Then the replication process always begins at specific sites, and these sites or these locations in the genome are called origins of replication. The two strands will serve as template, so the two of them will be a model for the copy of the newly synthesized strand, and they will be copied by complementarity. Keep in mind as well that the new DNA is synthesized only in the 5' to 3' direction, and this is because every new nucleotide entering in a strand must be added on the 3' end of the previous nucleotide. So as a result, if you look at a growing strand, you will see that the 5' end doesn't move, but the 3' end will be the one that elongates. So it is why we say that it is synthesized in the 5' to 3' direction. And finally, it is also important to understand that DNA polymerase is unable to add nucleotides on nothing. So it must have previous nucleotides added in order to be able to start the synthesis of DNA. So it is why we need to have another protein that puts in place a primer. So it is a little piece of DNA that will act as a sort of hook to start the synthesis of DNA, and this is the primase that will be involved in putting the primer. While in contrast, at the end of the chromosome, so when you finish the process of replication, what will happen is that there is a need for a specific process to replicate the chromosome completely, and this one will be achieved by the telomerase. So let's have a more precise view on the DNA polymerases. We call them polymerases because there are not only one, so there are many DNA polymerases. Here I give you two examples, one with the E. coli, or what is found most of the time in bacteria, and an example with mammalian cells. You see that we have several types of DNA polymerase. The ones that are the most frequently used for regular DNA replication are the ones that are surrounded by a red circle. And in mammalian cells, you see that it's the polymerase delta. It has, as the little plus indicates, capacity to proofread its own polymerization process. We will see in a minute what it means. And it needs to have a primer made of RNA or of DNA to be able to add new nucleotides. And it is located in the nucleus. And if you look at the other polymerases, like for example the polymerase gamma, it is a polymerase that is found almost exclusively in the mitochondrion. So it is involved in the replication of the mitochondrial DNA and not of the genomic DNA. Now, how does the polymerase delta work? It is a protein that has a structure a bit like a hand, so with things or parts that could represent fingers and thumb and the palm. You see that it is kind of folded, and it creates a sort of pocket in which the DNA will be able to insert itself. So the DNA will be kind of kinked when it enters in the DNA polymerase, and this will help with the process of addition of new nucleotides. So what does it require to synthesize DNA? First of all, you need a template because all the new entering nucleotides will be added on a complementarity basis. So the nucleotide that is added is chosen, so it can be a T, a C, or a G, but it is always chosen according to the nucleotide present in the template. So if in the template you have a T, then the nucleotide chosen for the addition in the growing strand will be a A. It also needs to have a 3' end or a OH available to be able to add the new nucleotide. And this is the nucleotide just before the newly entering nucleotide located in the same strand. Now, what happens in the polymerase is that the catalytic domain, so which is the domain that catalyzes the reaction of addition, will catalyze the formation of the bond between the 5' phosphate of the entering nucleotide and the 3' end of the growing strand. After this has been done, the DNA polymerase will proofread, so it has an exonuclease activity, so it is an activity, a capacity to remove a nucleotide, and this will happen if the newly entering nucleotide is a wrong one. So if instead of an A you have added a C, there will be a mismatch, and the exonuclease activity of the DNA polymerase will remove this mismatched nucleotide. So each time the polymerase adds a new nucleotide, it will also double check or proofread and remove it if it's not the right one. So when the synthesis occurs, you already know that, but it's always worth repeating that it will add a new nucleotide only on the 3' end of the growing strand. So the direction of growth is from the 5' to the 3' end. When the newly entering nucleotide arrives, it will have 3 phosphates added, so it is a nucleotide 3-phosphate. And the reaction catalyzed by the DNA polymerase will be the removal of the 2 phosphates at the end of the triphosphate, and it will use the energy released by the removal of the 2 phosphates in order to add the phosphate group on the OH end of the previous nucleotide. So it will make the previous nucleotide, then phosphodiester bond like OPO, and then the newly entering nucleotide stabilized in the growing strand. So we said previously that the DNA polymerase can proofread. How does it do that? It is relatively easy for the DNA polymerase to recognize that there is a mismatch, because a mismatch will in every case create a distortion in the strand. In fact, if you look at the geometry of two well-matched nucleotides, like for example a T and a A, or a C and a G, you see that the angle between the two nucleotides, the two complementary nucleotides, are of about 50 degrees. Angles of about 50 degrees when you look at the angle between the nucleotide and the sugar phosphate backbone. In contrast, when you look at the mismatched nucleotides, you see that the angle between the sugar phosphate backbone and the paired nucleotides is much bigger on one side and much smaller on the other side. So it is like 68 in case of a C matched with a A, and 46 for the A side. And if you look at the TG mismatch, it's also a big angle on the side of the T and a small angle of 42 on the side of the G. So this will be very different from the regular 50 degrees angle that you see in a normal well-matched nucleotides in a double-stranded DNA. Here, when you have a wrong match or a mismatch, the angles will be totally twisted when you look at the paired nucleotide compared to the sugar phosphate backbone. It will result in a distortion of the strand that will bring the newly synthesized DNA in a different direction compared to the normal flow into the DNA polymerase, and it will bring the mismatched nucleotide in proximity to the exonuclease activity of the polymerase, so the domain that is responsible for the exonuclease removal of the mismatched nucleotide, and this will remove the nucleotide and restart the synthesis after a well-matched nucleotide will have come in the strand. Now, how does the process of synthesis start? It will start in specific areas of the genome. These areas are always called ORI, and you will find ORI, or origin of replication, in every genome. It always contains a specific sequence that is called an autonomously replicating sequence. You can also hear the word consensus sequence. It means that it's a sequence that always means the same thing for the genome. So here in this case, the ARS, or autonomously replicating sequence, is a sequence that informs the cell that here will start DNA synthesis. In the human cells, you have many ORI containing ARS like that, and what will happen is that this sequence of DNA will be recognized by specific proteins. So the proteins are called origin of replication complex proteins, or proteins that recognize the sequence and bind to the sequence, meaning that here is an origin of replication. So these proteins will start to build up on the sequence and form a big complex that will open the replication fork. It will open the DNA double strand in order to start the process of further opening the two strands in order to then host the DNA polymerase and start the whole replication process. Now, among all the proteins present in the pre-replication complex, you will find the DNA helicase, and this protein will be able to bind with the double-stranded DNA, consume ATP, and use the energy released from ATP hydrolysis in order to break the hydrogen bonds between the base pairs, so that instead of a double-stranded DNA, you create two pieces of single-stranded DNA that will be able to serve as templates for the synthesis of new DNA. Note that the process of DNA replication will occur even if DNA is wrapped around histones, so you won't need to unwrap it. It will progress along the DNA and follow the loops around the histones. When DNA is opened like that by the helicase, it forms what we call a replication bubble. It means that you will have on a previously attached double-stranded DNA piece, you now have a part where the two strands are separated, and it creates an opening or a bubble shown here, and the replication process will start at both on the two sides and progress on the two sides of the replication bubble in what we call the forks, or the replication forks. Okay, so the left hand side replication fork will go on progressing at the left, and the right hand side replication fork will progress at the right, further opening the replication bubble and making it bigger and bigger. So with this, we have met the first requirement of the DNA polymerase, which was the requirement for a template. But we don't have yet the second requirement, that is the need for a primer. So we will find this need met by the addition of our RNA primer by a specific enzyme that is called the primase, and this enzyme has the capacity to start the synthesis of RNA anywhere on a template strand without the need for a 3' OH end previous to it. Okay, so the RNA primer here will be added by the primase. It will contain about 10 to 20 nucleotides, and it will provide the priming terminus, or the 3' OH end, that is needed for the DNA polymerase. So it will provide the second requirement, and that now the DNA polymerase will have both the template, which was the first requirement, and the 3' OH end, that is the second requirement, on which it will be able to add new nucleotide. So the DNA polymerization will always start with a little RNA primer that will provide the 3' OH end. So now that everything is in place, the replication process can start and go on. So this is what we will show you in this slide. It is a pretty complex slide, but we will go over things one by one. So first of all, we start with what we have seen previously, the DNA helicase that is always located in front of the replication complex containing several proteins. And this helicase will work in front in order to unwind DNA and make the two single-stranded pieces of DNA available for the DNA polymerase. When the helicase unwinds DNA like that, it creates two single-stranded DNA pieces, and one of them will serve as a template for a continuous synthesis. It is the leading strand, highlighted here and shown in grayish, while the other strand that we will see a bit later will have a synthesis that is discontinuous. This is why we say that the synthesis of DNA is semi-discontinuous. It is because one strand has a continuous synthesis and the other strand is a template for a discontinuous synthesis. So let's first have a look on the leading strand that is a template for the continuous synthesis of new DNA. Here you have the DNA polymerase that is well attached and maintained on the template, so on the single-stranded DNA of the leading strand. And this is maintained well attached by the clamp that attaches the polymerase on the single-stranded DNA. New nucleotides can be added on the 3' end so that every entering nucleotide will be added at the front of the complex, and the progression will work from 5' to 3', as we have always mentioned. So if you start the synthesis here, you will progress further and have a longer newly synthesized strand. So this one is pretty simple, and it is said continuous because it will go on like that up to the end of the DNA double strand, in fact up to the end of the chromosome or the meeting of a new replication bubble. Things are slightly different for the lagging strand, because the problem with the lagging strand is that if you follow the path of synthesis of new DNA, you would need to add new nucleotides on the 5' end of the growing strand. And we all know that synthesis is done on the 3' end. In other words, you add new nucleotides on the 3' end of the growing strand and not on the 5'. So if you look at the way the replication fork is moving, the synthesis of DNA is moving in this direction, while the DNA polymerase is moving in that direction. So it has to be reconciled because, in fact, the synthesis goes backwards while the moving of the polymerase goes forward. So in order to be able to do the two things at the same time, what will happen is that a relatively long piece of DNA will be uncovered, will be single-stranded. It will first be covered by proteins that will prevent its reannealing with another piece of DNA. And when you have uncovered a length that is long enough of the single-stranded DNA, RNA primer will be added somewhere in the loop. this RNA primer will be, in fact, the whole strand will be kind of pulled forward through the DNA polymerase complex and with the clamp, so that it reaches the polymerase complex, and from the 3' OH end of the primer, then the polymerase can start adding new nucleotides on the 3' end of the primer. And in fact, the strand will go on being pulled through the polymerase and the clamp while the polymerase can move forward. So basically, that's exactly what's happening. The synthesis goes backwards while the polymerase goes forward, but they are maintained attached in a loop so that you can do the two things at the same time, like this. And of course, once this fragment of DNA is synthesized on a long enough piece, it will meet with the previous fragment of DNA that had been synthesized in a previous round of replication, a previous round of looping like that. And these fragments of DNA synthesized discontinuously are called Okazaki fragments. So on the lagging strand, you have pieces of RNA that are primers, and you have pieces of DNA that are the Okazaki fragments. Eventually, later, what will happen is that the polymerase will replace the RNA primer in order to make a continuous strand of DNA. So this is a relatively complex process, and because it is difficult to show in a fixed medium, I added two links to two different videos that you can find on YouTube, and that show the process in real time. It is sometimes easier to visualize it when you have a real animation. So once the lagging strand has been synthesized, when the Okazaki fragment merges with the previous Okazaki fragment, it will need to be ligated so that it creates a continuous strand and not small pieces that are left unjoined. Okay, so the ligase is very important in order to make a strand that is with no holes in it. Okay. And the first origin of replication that we had at the beginning will always be found in the middle of the bubble. But if you look at the bubble itself, you see that it has neighbors. So you have several bubbles, several origins of replication on the same chromosome. This is true for eukaryotes. In fact, for prokaryotes, they usually have only one, but in eukaryotes, you have several origins of replications, so several bubbles of replication, with the forks moving in both directions, so in opposite directions. Forcefully, the bubbles of replications will merge into a single bubble of replication, and eventually it will merge with the next neighbor bubble of replication, and the same origins of replication will now be found as neighbors on a single newly synthesized chromosome. Now, there is another part of the chromosome that is a bit problematic, and it is the end of the chromosome, or the telomere. So the problem with the telomere is that when the DNA polymerase arrives in this area, eventually, after a moment, it starts to have not long enough of single-stranded DNA to be able to perform the whole replication process. And when the strand becomes too short, it will simply destabilize the polymerase, and the polymerase will fall off the template strand before the synthesis of new DNA is completed. So it will always leave a little part that is single-stranded and not fully synthesized. Of course, a cell could not leave it like that, because otherwise at each round of replication we will have shortening telomeres. So instead of that, there is a protein called the telomerase that will prevent the shortening of telomeres at each round of replication. It still does happen quite a bit, but cells that have to replicate relatively often will have a telomerase, and it is one of the features also of cancerous cells that often have increased telomerase activity to prevent the shortening of the telomeres. So how does the telomerase work? It is a protein that is able to bind to the single-stranded DNA found at the telomere. It has in it, inserted into the protein, a RNA primer or a RNA template that will be used for the synthesis of new DNA on the 3' end of the template. So what will happen is that the RNA will serve as a template and DNA will be synthesized on the 3' end of the telomere, and it will move forward so that you have an increased length of the DNA template. So you elongate the template of the telomere artificially. In fact, it creates more, it increases the amount of nucleotides that are found at the end of the telomere. Then it provides a longer telomere, a longer single-stranded telomere that makes it possible for the DNA polymerase to make the synthesis of this DNA and merge with the previously completed DNA double strand. Again, there will be a little part that will remain single-stranded, and this little part will be removed after by exonucleases. So you transiently elongate the telomere so that you provide a longer template, and the synthesis can be achieved by the polymerase because of the longer template, and the little part that is not synthesized will be removed after. It is how cells can go on with the replication of their DNA without shortening their telomeres too much. So if we do a little summary of all the proteins involved in the replication process, we will cover the bacterial and the mammalian proteins. They are shown at the same time on this slide, but the first one always corresponds to the bacterial and the second one corresponds to the mammalian protein. So ahead of the fork of replication, so in front of the fork of replication, you will have a protein called gyrase or topoisomerase 1 and 2 in the mammals, gyrase in the bacteria. And this is a protein that releases the tension of the DNA when it is supercoiled. So it will work really ahead of the fork in order to prevent knotting in coils, supercoils in the DNA in front of the fork. Into the fork, you have in front the helicase that will unwind DNA and separate the strands. You will have then the single-strand binding proteins or RPA in mammals that will cover the lagging strand template. You will have the primase in both the bacteria and the mammals that synthesizes the RNA primer. You will have the beta clamp or pCNA in mammals that attaches and releases the lagging strand to the polymerase each time the loop is formed. And then you have the DNA polymerase 3 or DNA polymerase delta and epsilon in mammals that synthesizes both the leading and the lagging strand, always in the 5' to 3' direction when we consider the newly synthesized DNA. Finally, behind the fork, you have the DNA polymerase 1 or FEN1 in mammals that will remove the RNA primers and replace them by DNA, and the DNA ligase that will link the fragments, the Okazaki fragments, so that you form a continuous lagging strand. And at the end of the chromosomes, you will have the telomerase that is found, of course, only in eukaryotes because prokaryotes have a circular chromosome, so you don't need to have a telomerase in prokaryotes. And the telomerase will prevent the shortening of the telomeres. So as you can see, this process, even if simplified here with only the main and most important proteins, is a process that is extremely well regulated and tightly controlled. So how efficient is this? First of all, the quantity or the rate of errors or the rate of wrong incorporation of nucleotides during the replication is called the spontaneous mutation rate, because it is the number of mistakes that the polymerase will leave in the newly synthesized DNA. Now you remember that the incorporation of a particular nucleotide, so the choice of a A versus a T in the growing strand, always depends on the geometry of the base pair. So a T will pair with an A because of its geometry. Now, because of the geometry as well, a DNA polymerase will be able to double check or to proofread and see if the geometry is wrong or if it is twisted compared to the normal angle that it should have. So it allows for a careful selection of the nucleotide plus the proofreading of the nucleotide that has just been added. So overall, it will leave a very low number of mistakes at each replication round, and this low error rate is about 10 to the minus 9. So it means that you will have one mistake per billion of nucleotides. In addition, because of this proofreading and careful selection in the same molecule, in the same DNA polymerase, it allows a very low error rate while still allowing a replication that is relatively quick, because the DNA polymerase is able to add about 1,000 nucleotides every second. So you see that it's a pretty quick replication rate, and still a replication rate that has a relatively low error rate in the replication process. So this closes the chapter about DNA replication. The next video will cover DNA repair and damages.